We will continue our discussion about the effect of this stiff matrix on the pancreatic cancer cells to develop the chemoresistance against the drugs that are designed to kill the pancreatic tumors. In the previous video, we have discussed that with the increase in the stiffness, there is an increase in the chemoresistance. In this video, we will discuss the long-term effect of these pancreatic cancer cells when they are kept in this stiff matrices and compare it with the soft matrice. And we will check the chemoresistance that are developed through the use of different drugs that are designed to target these pancreatic cancer cells. So panel A is about their plan, how they have planned their experiments. So they prepared the organoid, and before using this gemcitabine treatment, they grow the organoid for 3 days till it acquire organoid will acquire a size of like 50 to 75 micrometer. And same things, and they did these things for two different conditions. First one is this help low, and second one is this help high. Help high, you can consider it as the stiff matrices, and help low is the low matrices. And when they check the effect of gemcitabine concentrations to this cell toxicity, what they have find out is that there is not much difference they have observed during the first passage, and they check the effect of development of chemoresistance at one passage and at four passage. And in the one passage, they do not see too much difference between help low and help high. However, in the fourth passage, they see that difference between the help low, the chemoresistance developed in the help low matrices and chemoresistance developed in the help high matrices. The quantifications are shown in the panel C, where they have quantified based on this IC50, which is considered as the 50% of these drugs required to inhibit, not 50% of these drugs, the amount of drugs required to inhibit 50% biological characteristics of the pancreatic cancer cells, biological functions of the pancreatic cancer cells. So they use this IC50 to check the amount of gemcitabine required for this IC50, and they have find out is that in the case of help high, at IC50 levels are statistically significant compared to the other case, and in the help low at the passage 4 there is not much statistical difference observed. And they did the same experiment with the single cell also, and in the single cells they check the results, they check their effect of this effect of different matrices, different stiffness of matrices on the development of chemoresistance in pancreatic cancer cells, and they checked it at passage 1 and passage 4 on day 6. After day 6, and at the passage 1, they do not see too much difference in the help low and help high in terms of this cell viability with respect to the gemcitabine concentration. However, at the passage 4, they clearly see the difference that in the case of help low, there is a in the case of help low, the cell viabilities are low, whereas in the case of help high, the cell viabilities are high, indicating that the higher stiffness is more favorable to the pancreatic cancer cells. The quantifications are shown in the panel F, where we can see that in the case of passage 4 in the help high conditions, the IC50 values are statistically significant compared to the other 4 cases. In the case of help low at passage 4, the difference is not much statistically significant compared to the help low in the passage 1, but it is significant compared to the help 1 and high matrices or high help at passage 1. After validating their results, and are in line with the previous studies, they also wanted to check its impact on different or some other drugs that are available commercially. So they check their results on this paclitaxel's IC50 also, and also on this FOIIC50 on the single cells at passage 4, and they have seen the similar results that in the case of high help matrices they see that the IC50 values are statistically significant in both the cases compared to the low help matrices. Thereafter they wanted to check Thereafter, they checked the in vivo analysis in the pancreatic cancer in patients who are affected with the pancreatic cancer. So they analyzed two different patients in HELP low and HELP high conditions, two different patient tissues in HELP low and HELP high conditions. And what they have find out is that in the case of HELP high conditions, they can see that the tumors are growing, whereas in the case of HELP low conditions, the tumors are low compared to their respective HELP high conditions, and the results are quantified in the panel J and panel K. The panel J is about patient number 2, where they did the experiment on the single cells. And what they have find out is that cell viability are low in the HELP low conditions, where the cell viabilities are high. Where cells, I mean the pancreatic tumor cancer, pancreatic tumor cells, the cell viabilities are high, and these things are quantified in this gemcitabine IC50 analysis, where the high matrices required the more gemcitabine IC50 compared to the low matrices low help matrices required a statistically reduced amount of gemcitabine IC50. And similar results they have observed for this patient 3 also, where uh, where the where the HELP high conditions has high cell viability compared to the HELP low conditions, and this thing is quantified on the right hand side, where we can see that high HELP matrices required more IC50 compared to the low HELP matrices, or cells that are grown in low HELP matrices. So I hope you got an idea that from this whole figure we got an understanding that with the increase in the matrix stiffness over a long period of time will cause or will develop chemoresistance against the drugs that are designed to kill these tumor cells. If you need to understand more about their experiments in detail, or if you need to understand their if you want to check the supplementary data, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.